Hello everyone, good afternoon, ladies and gentlemen. Myself, Dr. Satish, working as a plant breeder in ITC Limited since 15 years. As a plant breeder, I am looking for development of new varieties and hybrids for respective growth regions of our organization. So before going in deep about this today's topic, the role of pre-breeding in crop improvement program. So my overall flow of presentation will go in this fashion. Next 30 minutes, I will take you through what is pre-breeding, followed by genetic enhancement, and fundamental difference between the pre-breeding and commercial breeding. And what is the pre-breeding purpose and requirements and events, and I sum up with conclusions. So what is pre-breeding? Pre-breeding means transfer of desirable characters from unadapted materials to the intermediate population and same population used by breeder in developing the new varieties. It's like a bridging between the breeders and genetic resources. Then comes to the what is the basic concept of the pre-breeding. The basic concept of the pre-breeding is integration of desirable genes from the different kinds of germplasm into readily available material with less linkage drag and exploration of variation in many ways, and finally, and improving the genetic diversity by using wild relatives such as resistant to different stresses. Then there are various methods are there, but exploitation of cultivar improvement is limited by various factors like cross incompatibility barriers and linkage drag. But when it comes to pre-breeding activities using promising land races, wild relatives, and different cultivars which are popular in respect to crops and which are developed by different institutes by using the different gene pools, crops like wheat, chickpea, PGNP, and groundnut, so that they have wide adaptability and broader genetic base. And at the same time, availability of markers, they are great in helping reducing the linkage drags and as well as improving the efficiency of integration in pre-breeding programs. This is the basic concept of the pre-breeding before you go into the actual varietal development. And in addition to this pre-breeding is not significantly different from the overall framework of plant breeding or varietal development program. This is one of the first step or previous step for sustainable plant breeding. Why I'm saying sustainable plant breeding means whatever the variety or hybrid you have developed, that should be stable over the years in different growth regions. So that's why we should, pre-breeding is mostly useful for sustainable plant breeding approach. And this pre-breeding population consists of materials which are useful characters and capturing its genetic diversity and putting those all the useful traits into the improved cultivar, and simultaneously this pre-breeding leads to the overall value addition in terms of overall germplasm pool. Why this pre-breeding is required? Why this pre-breeding? Look at these different crops which are cultivated across the globe. When it comes to potato, there are 4 varieties which occupies the 75% of the cultivated area. Similarly, in maize, only 6 varieties are occupied the 70% of the cultivated area. When it comes to major crops like rice in India and especially in the Asian countries, which occupies the 65% with 4 major varieties. Similarly, in pulses like soybean, that 6 varieties occupies for the 50%. And finally, that one more cereal crop, which is wheat, is actually occupies the most of the globe when it comes to other crops, which is occupied by 50% of the cultivated area with 9 varieties. Why? If these 6 limited varieties means what will be happen? Because of the limited variety, there will be narrow genetic base, so that because of the narrow genetic base, it leads to major diseases and pests. Let's go through the what happens. What is the consequence of the narrow genetic base? I'll take you through the next few slides. As I told earlier, this increased narrow genetic base has so many setbacks in our history because it leads to different disease and pests. If you look at back the history in 1845 and 46, the major famine is called this Irish famine, it results in 50% loss in the potato plantations because of the disease late blight of potato. Similarly, in 1869, coffee rust, which entirely wiped out the plantation in countries like Ceylon, and that entirely replaced the coffee with the tea because of it is because of narrow genetic base, and we are unable to replace with the better cultivar. And when it comes to 1943, that brown spot of rice, which is called Bengal famine, it results in more than 50% loss of the rice crop in countries like India. And followed by 1970, that major disease in corn, that is US corn blight. This is an epidemic, and it affects the most of the corn from the Midwest region of the US. And there is almost range of 50 to 100% loss in the entire corn fields of US Midwest region. What it shows, what it example shows, what is all these famines told that because of limited entries, because of limited variability, because of limited varieties, because of their not exploited fully what are the available germplasm we have, it leads to lot of uh, vulnerable to insects and pests. So that there is a call for to improve the genetic base, and we should shift from narrow genetic base to broad genetic base. And especially in crops like wheat, that continues in case of wheat. Generally, if you see the in case of wheat stem rust is historically the most damaging disease of wheat. The disease has the capacity to turn health looking crop only weeks away from the harvest into nothing more than a tangle of black stem and shriveled grains at harvest. Hope you're looking these diagrams. Right side the things that below field is uniform and healthy harvest field, and within the weeks how it was turned into, see the stem rust kind of symptoms, into nothing more than a tangle of black stems and shriveled grains at harvest. Under suitable conditions, yield loss of 70% or more are possible. Wheat stem rust is highly mobile, spreading rapidly over large distance by wind or via accidental human transmission, like infected clothing, one second, infected clothing or plant material. When stem rust has largely been under control of over 3 decades due to the widespread use of resistant cultivars. In 1999, a new virulent race of stem rust was identified from wheat fields in Uganda, popularly known as a UG99 after the year and country of discovery. Using North American scientific nomenclature, UG99 is known as a race TTKSK. UG99 is a cause for concern as it exhibits the unique virulent patterns. No other race of stem rust has been observed to overcome so many wheat resistant genes, including the important gene SR31. By 2007, this race, UG99 race, had spread via wind movements out of East Africa to Yemen as far as Iran also. Rust pathogen challenges rapidly, often by mutations. 6 additional variants are now recognized in the UG99 lineage. All exhibits an identical DNA fingerprinting, but very much difference in virulence pattern. And additional important resistance genes like SR24, SR36 have now been repeated by the variants of the UG99. So UG99 are all the variants of UG9 considered as a major threat to wheat production, with an estimated 80 to 90% of the global wheat production. It shows that how it has spread within the weeks, within the plant is ready to harvest. So that this threat continue, and this is the point we need to concern much about how to address such kind of things. So finally, what I like to tell you, there is a time to improve the genetic enhancement. Right, so that in crop improvement program, narrow genetic base resulting in a slower progress, as well as a increased risk of genetic vulnerability, as we witnessed in the different crops like wheat, potato, corn, and coffee. This results in the accumulation of unused potential germplasm, which are in our kitty. In order to witness or break these bottlenecks, we should come up with some good gene pool so that we can enhance the value of germplasm by utilizing by doing the genetic enhancement or pre-breeding methodology. There is a minor difference between the genetic enhancement and traditional breeding. Just this genetic enhancement it does not include any varietal development. It refers to the only improvement of the available germplasm in our kitty. So that once you enhance the germplasm, that can be more readily used in breeding program for developing a different varieties and hybrids for the different crops as and when required as per the growth regions. Followed by next one is what is pre-breeding and commercial breeding. So pre-breeding is, as you mentioned in previous slides, it is like genetic enhancement of germplasm. But in commercial breeding, it leads to the development of productive cultivars. Productive cultivars means new varieties and hybrids which with the good traits. And this can, what happens, pre-breeding generally broadens the genetic base, whereas a commercial breeding, it can narrow the genetic base by repeated use of same material in our breeding program. But when it comes to in terms of duration, pre-breeding is a very long-term program. But in commercial breeding, because of pre-breeding activities, we can adopt our commercial breeding program medium to short term. Then the chief methods we are used for this producing the pre-breeding materials is one is backcross method of breeding, then marker-assisted breeding, and genetic engineering. Whereas a commercial breeding that depends on the method of pollination and fertilization, all breeding methods were followed. But pre-breeding methodology usually followed by the public sectors in countries like India. But whereas in commercial breeding, both public and private sectors, and they participated very vigorously to develop the new cultivars, which so that they can feed the farming community to increase their cost-benefit ratio. Then what are the activities in pre-breeding? See, what is the fundamental difference between the pre-breeding and post-breeding? This commercial breeding is, pre-breeding is very long-term process. Commercial breeding depends on the pre-breeding activities. We can fine-tune our varietal development program into medium term or long term. When it comes to pre-breeding, first we identify the germplasm. Among the germplasm, the best one we can select and we evaluate for different traits like abiotic stress, different biotic stresses, and pest and disease control, and as well as to improve the nutrition quality like grain quality. Once we develop this platform, and we will make some genetic analysis, and you can come up with some different markers. And similarly, we can develop different genomic tools, and we can come up with the better gene identification, so that means straight away we put into our commercial breeding program. And overall, what happens, we can develop a good platform so that we can use for as and when required. This is overall we call as a development of germplasm. So that pre-breeding is once again I am repeating, pre-breeding enhancement of the available germplasm to give it for the commercial breeding program. Once we develop the germplasm and once you transfer to commercial breeding program, we can develop the best varieties based on the trait interest and by using the different marker-assisted selection breeding methodology also. This is overall the fundamental difference between the pre-breeding and commercial breeding. So in addition to this, what I explained in previous slide, generally pre-breeding start with plant genetic resources. We will collect the different plant genetic resources like different germplasm from the different crops, and we should maintain and we will characterize all those lines as per the parameters. We know there are different parameters based on the crop. We call it dust parameters because it should be distinguished, it should be uniform, and it should be stable over the existing lines. And we can characterize based on that. We will identify the parental lines, and we will develop the parents. And same time we can evaluate the best parents or best plant genetic resource or germplasm, and we can develop the variety also, so that variety will release in that fashion. But These plant genetic resources all will be stored in gene banks. As and when required, we can, based on the our trait of interest, we can select those lines from the germplasm gene bank, and we will develop we will use all the pre-breeding methodology, and we can later transfer to commercial breeding program, and we come up with the new cultivars which can better than the existing cultivars and which can throw the more yield and quality traits to the farming community. So that like this, it was happened and benefited by the farming community in crops like rice, especially in wheat, corn. So that pre-breeding is it's like a bridge. It connect all the plant genetic resources of particular crop and our plant breeding program in crop improvement division. So that genetic resources are like the cultivated type germplasm. There are various kinds of germplasm, but we should choose the cultivated type germplasm as well as wild relatives, because wild relatives contains a very broad genetic basis, so that we can capture and useful in our useful, and we can capture it into our existing cultivar development, followed by pre-breeding program, so that whatever the identified in our germplasm, we should evaluate, and we can identify the promising donors based on our evaluation, and whatever the evaluated, will identify the better materials as a parents, and we made the hybridization between the adopted lines, and we should come up with some pre-breeding population which should be uniform. This all methodology, all the programs, all the steps will take place for almost 6 to 10 years. And after that, we will transfer all our pre-breeding materials to the core crop improvement program. There are various working collections, and we will make severe cross combinations, and we can develop the good recombinants, and what are the best recombinants which can deliver good yield over the existing cultivars in the particular region, and we can release that particular variety as hello.
Yes, Dr. Satish, we can hear you.
Is it? Yeah. Is it
Now we can hear you.
audible?
For a few seconds,
Okay.
your voice was not coming.
Yes, ma'am. I think there are some technical glitches.
Yeah, please continue.
Shall I continue?
Yeah.
Okay. So pre-breeding is like a bridge between the different genetic resource and crop improvement program. So, so what is the purpose of pre-breeding? Main purpose of the pre-breeding is, as you know, we should convert from the narrow genetic base to broad genetic base, and to break the yield barriers, and as well as to develop a donors for different stresses and a tolerance, and to develop donors for the different avoided stress and tolerance, and to improve the quality of the products, and finally to improve the variability over to develop good agronomic characters. These are the purposes of basic purposes of pre-breeding program. And when it comes to crop wise, the main purpose is to broaden the genetic base and to improve the variability of the population. To break the yield barriers, there are various examples in different crops like wheat. Actually, there is a synthetic hexaploid wheat derived from the durum and thassi, and this synthetic hexaploid crossed with the local cultivar, and it can increase the yield up to 20 to 35% in countries like China. And in Kabul chickpea, we with the use of pre-breeding materials, we can develop a lines for terminal heat and drought tolerance. And in chickpea, we can develop a lines resistant to ascocita blight. In lentil, we develop a lines which are tolerant to drought and heat. and as well as different diseases like stem phylum blight as well as root rots also. And similarly, Agilops, by using different species, we have developed different resistant lines for stress resistance, for biotic stress resistance. And similarly, for abiotic stress resistance in synthetic wheat for drought tolerance, and by using chromosome manipulation W4909 and W4910, by using these two genes, AJDAJ5 in additional chromosomes from Thynopyrum gentium. By using the chromosome manipulation, we developed the donors for abiotic stress tolerance. And NAX1 and NAX2 tolerant genes from the Triticum monococcum to develop a salt tolerance durum wheat. And when it comes to the source of quality, grain legumes genetic enhancement in case of chickpea and arhar, and various synthetic exoploid lines used to improve the quality of the wheat products in case of Triticum turgidum and durum, and has leads to 20-30% more iron, magnesium, and zinc. And by using the chromosome link of 1B by 1R translocation to produce high zinc efficiency cultivars to improve the quality of high zinc in case of wheat. And finally, we should major aim of any breeding program is to develop a variability or to create a variability to improve the other agronomic characters. And finally, the pre-breeding for climate smart grain legume crops in global level in relation with the respective country climate change. Then what are the requirements of pre-breeding? We must have a genetic resources. All the materials are available for improvement are for the cultivated plant species. And whatever the genetic resources we have, we must characterize and we evaluate based on the dust characters. And as well as because we are transferring good materials to the existing cultivars, one must have a local regional growing regional knowledge and diversity of the population of the particular crop, and same time knowledge on the interspecies of the particular genus, and as well as the relationship among those species is also highly required to improve the diversity of the particular field. And finally, what are the events for breaking yield barriers and utilization of genetic diversity so that more diverse means we can come up with a good yielders or good pipeline of varieties so that we can distribute the different cultivars based on the geographical region. And we can develop a different morphological and physical idiotype based on the growth regions, and improvement of early generation selection criteria so that if it is short duration, medium duration, long duration means we should define the duration of the variety based on the flowering pattern, and we can develop varieties based on that particular growth region. And finally, when it comes to varietal development point of view, in this we must have a knowledge on its genotypic environment management interactions, so that only genotypic environment interaction is sufficient for the estimation of phenotypic efficiency. But to know the better understanding of the crop of particular region, management is also one of the crop management is also one of the parameter which is highly required to adaptation of the good variety. And as well as one must have a improved understanding of the particular crop phenology and its development, so that broadening of genetic base of variety we can develop by use of winter and spring hybridization, and we can import the germplasm from the other center of origins, and we must develop a different plant type approach, and by using alien species for biotic and abiotic stresses based on the chromosome manipulations, and exploitation of heterosis for hybrids. Exploitation of heterosis hybrids. Hello.
Yes, Dr. Satish.
Is it audible, ma'am?
Yeah, now you're audible.
Yeah. So when it comes to breaking yield barriers for different crops, in this exploitation of heterosis means the F1 offering whatever you're getting, it should be better than the existing parents. For this, we have, in case of often cross pollination like pigeon pin tobacco, we use the CMS system to develop the hybrids. In pigeon P, this ICPH1, in tobacco, CH1 and CH3, they are the first hybrids with developed with the CMS background in countries like India. And when it comes to different interventions from the biotechnology side, like gene pyramiding, it's like pulling off all the useful genes into one block. And we should develop a markers selection for to deliver a variety to resistant to the different biotic and abiotic, and to improve the different quality traits. And finally, structural and functional genomics by using the different bioinformatics tools, and we should develop by pre-breeding approaches, what are the materials you develop from this methodology. And abiotic stresses, because abiotic stresses because of climate change, we should address heat and drought, salt stress, as well as different water stress conditions like right from high water condition to water lagging condition. Hello? Hello, ma'am? Hello, ma'am? Hello, ma'am, is it audible? Hello? 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 Yeah. There are certain aspects of pre-breeding which can break the yield barriers. In case of chickpea, it can, by using the different pre-breeding materials, we can break the terminal heat, and drought tolerance varieties we can develop, and resistant to ascocita blight. And donors have been identified for a range of traits like botrytis gray mold, nematode resistance, yield traits, fusariumil resistance, and finally drought or salinity tolerance. In case of lentil, tolerance to drought and heat, and resistant to stemophyllum blight, root rots, heat tolerance, earliness, and because of for its large seeds, and for different diseases like rust and powdery mill resistance, and for different nutrients improvements like iron and zinc, and for different yield trails, and as well as fusarium and drought salinity tolerance.
Hello.
Yeah, ma'am. Is it
Yeah,
audible, ma'am?
it is audible, and so your presentation is completed.
Just uh, just 2 minutes, ma'am.
Okay, okay, okay.
Hope it is visible now, ma'am. My slides.
Yeah, it is visible. I can see the slide
Hello,
which is.
ma'am.
I can see the slide which is written as pre-breeding. That slide I can see.
Madam, there it was stuck. I think I'm continuing. I will share, ma'am.
You can
Just a
stop and
minute.
restart. Stop and
Yeah,
restart
ma'am.
sharing.
Sure, sure. Is it visible, ma'am?
Yeah, it is visible.
Yeah. Then what are the basic steps in pre-bidding these? What is the purpose of pre-bidding is to broaden the genetic base and to break the yield barriers, to develop a donors for biotic and different biotic stresses, and to improve the quality and to improve the variability of other agronomic characters. And as I told you earlier, there are different this one with some I will quote some of the examples. As I told, the major this one is to improve the broaden the genetic base and to improve the genetic variation. In case of hexaploid wheat, we have developed synthetic varieties so that whatever the developed synthetic which was crossed with the local cultivar and we can increase the yield up to 20 to 30% in China. And in chickpea, we have developed different lines which are resistant to terminal heat and drought tolerance. And this is like ascida blight. And in lentil, we have tolerant to drought and heat, and even resistant to different diseases like stem phylum blight and root rots. And even edgelops, we're using different species diversity, and we can develop a different genes for the lines for resistant to biotic stresses. And similarly, in wheat, we have developed different lines uh, to resistant abiotic stresses by using different genes like W4909, 4910, and to by using the additional chromosomes, alien chromosomes kind of categories, and NAX1 and NAX2, which are highly tolerant to salt, salt tolerant varieties, and we have developed in durum wheat in this case. And by increasing the source of quality genes in grain legumes and in synthetic hexaploid lines by utilizing the 1B1R translocation I gene chromosomal link. And as well as we know the major objective of this pre-breeding is increased variability to improve the other agronomic characters also. And as well as by utilizing pre-breeding approaches, we can develop a climate smart resilient varieties depends on the country of the development of the particular crop. And what are the basic requirements? Genetic resources and its characterization, evaluation, and the diversity of the resources, and as well as their interrelationship between the species and how they're behaving over the years. And what are the events for the breaking yield barriers in case of pre-breeding approaches, and by utilizing genetic diversity and development of different idiotypes of morphological and physiological and improvement of early generation selection criteria, and as well as in addition to G into E, we can add management interactions so that region specific crop development activities are possible, and improved understanding of the crop phenology and its development. And finally, the broadening of genetic base of the varieties is possible through the winter and spring hybridization and use of acquiring the germplasm from the center of the origin where it was developed. And we can always look for the different plant type approach and use of different alien species for by using for biotic and diverse stresses, and as well as exploitation of heteroses for development of hybrids for the various methods. But in often cross-pollen crops like pigeon pea and tobacco, by using the CMS system, we have developed the commercial hybrids and various biotechnology interventions like gene pyramiding, marker selection, and structural and functional genomics. And as well as by pre-breeding approaches, we can address the abiotic stresses by heat, heat and drought, salt stress, and waterlogging. And in case of chickpea, we have developed the varieties, developed donors for Botrytis grow mold, nematode resistance, and we developed yield traits entries and fusarium resistant varieties and drought or salinity tolerance entries. And in case of lentil, in addition to that, we have developed it earliness of the variety and resistant to different diseases and improving the nutrient quality index, so iron and zinc, and as well as resistant to different diseases like fusarium. And similarly, drought and salinity tolerance entries also be developed by utilizing the pre-breeding approaches. And in case of wheat, we can broadening the genetic base of the variety or by using the winter and spring hybridization and exploring the germplasm from the where it's sourced. And in addition to this, various methodologies followed in other crops that also we can utilize in these crops and we can develop. And in case of other crops, And the genome mapping and synteny of gene sequence from the wheat and barley. Synteny means generally students of genetics will use so that similar kind of loci on the same chromosome, so that it leads to genetic called linkage drag. So it is very difficult to break that lags by using different tools of pre-breeding, and we can decode that one, and we can develop a varieties for different evolved stresses and tolerance, and evolved stress and tolerance, and utilize it for the other crops. And phenotypic characterization with respect to its climate change to improve the varieties of barley, especially in the case of heat water use efficiency. And in case of groundnut, we should develop entries for the genetic enhancement for grain yield. And these are the major global research doing at the global level to break the yield barriers. Generally, in case of Kabuli chickpea and lentil, it is doing at the Icarda Syria, ICRISAT, and IIPR Kanpur, and exploring the different resources of wheat and barley for genetic discovery for developing a variety in climate change. And the institutes like IRI, NBPGR, IIPR, New producing relative varieties by using pre-breeding approach. And pulses like soybean production research by breaking yield barriers by using the pre-breeding entries. And these are the collaboration of different institutes like ICARDA, ICR India, especially when it comes to wheat, directed of wheat research, kernel is involved in this one, in addition to the CIMET Mexico. And in case of chickpea, the ICARDA Syria and the leading pulses institute in India, Indian Institute of Pulses, which is Kanpur, was involved to develop the donor traits for the botrytis gray mold, nematode resistance, for different yield traits, fusarium resistance, and drought tolerance. And in case of lentil, again, ICARDA Syria, and again, this same pulses institute was involved, supported by ARI New Delhi and NBPGR New Delhi. And they have developed traits for tolerant to drought and heat, and resistant to stem phylum blight, root rots, and to earliness to produce the large seeds, and resistant to different rust and powdery mealies, and to improve the iron and zinc content, and as well as to develop a resistant to fusarium wilt diseases. With this, what I can conclude that pre-breeding activities, they can utilize the wide relatives, which gives the high priority for breaking the yield barriers, so that one must focus on the identification of traits so that they can develop for the new climate resilience cultivars development with a broad genetic base. And we should always enhance the gene pool through the genomic assisted pre-breeding, and this genomic assisted pre-breeding will overcome the linkage drag, and that will facilitate the transfer of useful from the wild relative to genetic enhancement of the different legumes than the conventional method. Because conventional method means set of genes will come along with the required gene, but when it comes to genomic assistant, by using of genomic assistant tools, we can get the exact gene what you want to facilitate for the transfer of the useful genes. And same time stabilizing the yield and higher productivity under the biotic and diverse stress environment. And seeds of the appropriate cultivars agri farmers for using the various cropping system would be available for the multiplication and distribution. And wild range of variety will be available to farmers, so that broad genetic base and more genetic diversity will exploited, so that they cannot exploit to any deadly diseases or pests. With this, I can very much thankful to Agricultural Information, and very much thankful to giving this opportunity. Thank you. Hello, ma'am.
Yeah, can you hear me?
Yes, ma'am. Now it is hearing.
Okay. Yeah. Okay. Yeah. Thank you for the detailed presentation. Now I'll take up some questions.
Yeah, ma'am.
So, first question is, what are pre-breeding approaches and how do they contribute to sustainable crop improvement?
Yes, ma'am. Actually, as I told in my presentation, there are different approaches. Like say, if you want, first one is improvement of the genetic base, so that we should identify the different variability in the germplasm, What are the variability in germplasm? We identify the better parents, and we can make the hybridization among the better parents, and we will develop the better lines. And so that we are using wild genomic, wild gene bank or germplasm lines, so that we have a varied diversity, so that it can withstand any unprecedented pest and diseases.
Okay. Next question. How can pre-breeding techniques help farmers in improving the yield and quality of their crops?
Yes, ma'am. Actually, this pre-breeding approach itself is using of the native cultivars, wild relatives, because they are actually good at because of vigorous crop improvement or plant breeding program. We are confined to only limited number of entries. If you done at the farmer level, or where it's like hotspots are there, where if we cultivate or we develop, we have wider variability, and we should use that variability for different traits to improve the yield as well as quality, or what is the trait of interest for the particular region. So that it's always better to adopt the entries, not existing entries, which are not unadopted varieties. So that pre-breeding itself tell you that we should utilize the unadapted varieties into intermediate population. What we are doing, that we convert into good cultivar.
Okay. And what are the key challenges faced by farmers and agri entrepreneurs in crop improvement, and how can pre-breeding approaches address these challenges?
Yeah, ma'am. Nowadays, everyone is every farmer or every asking uh, hybrids, because they throw some hybrid vigor. Hybrid vigor means one side we are saying that our whatever the varieties are hybrids developed, they're throwing good hybrid vigor. But same time, cost of cultivation is one side is escalating. So that our pre-breeding program is not only for only improve the yield and quality, should aim for improving other agronomic characters, so that simultaneously cost of cultivation should comes down. Otherwise, at the end of the day, profits, even though our varieties delivers high yield, if cost benefit ratio or profits is like previous years, there is no meaning in delivering the good varieties. So that in addition to developing a good variety, we should adopt a good agronomic practices, and we should identify varieties with minimum inputs and minimum with higher population. The whatever the or minimum population, whatever the variety in particular crop can develop give a better yield, we should adopt that kind of varieties, so that that will reap a good benefits to the farmer.
Okay. And what are some examples of successful pre-breeding projects that have resulted in improved crop varieties?
Yes, ma'am. In case of wheat, especially, because if you see the wheat, there is a especially now very narrow genetic base, ma'am, because you will keep on using the same varieties. But what happened, ma'am? They're susceptible to lodging. So in order to break that one, we should again go back to our old gene bank, and we should identify the again we characterize, or we can evaluate all the lines. Definitely there should, this is proven also, ma'am, in case of wheat in India. So that we should identify the lines which are resistant to lodging. That trait of interest we should transfer into our existing cultivar, which is giving good yields by using a backrest method of breeding, so that it can give the same yields and provided resistant to that lodging kind of symptoms, ma'am, during unprecedented weather conditions. This is proven in case of wheat, ma'am. Of course, in pulses also, a lot of examples are there. So that we can improve the yield traits as well as the resistant to different. Just a live example I mentioned in wheat, but there are in pulses, we can transfer we can increase the size of the pod by using the old germplasm, through the pre-breeding methodology.
Okay. And how can students interested in agriculture and crop improvement benefit from understanding pre-breeding approaches?
Yes, ma'am. Actually, straight away, pre-breeding is a bridge between the germplasm or genetic resources and crop improvement. So that one who wants to pursue their interest in this domain, so that first they must know the what are the wild relatives, what are the different kinds of variability available in countries like India, and they must study. The rest of process is it's very easy. So that first initial that prior step is identification of germplasm lines, and continuation of pre-breeding methodology is the basic step. So that can add more value. If you are one guy is good at molecular breeding, he must have a hold on the its varieties, its germplasm, how it performs. Then he can transfer in with less period of time, whatever the trait of interest is required. It's highly useful for the students who are interested in this domain. And it's highly interesting also. It can throw a lot of insights. Actually, while we are working, only we are working for productivity and quality. But while we are working, we can throw a lot of insights and we can observe so many other characters, like how it behaves, if it is a high rainfall, if it is high moisture, if it is low moisture, and how it can withstand if there is any biotic and stress or happen during the course of our experimentation also. That all will we need not to do separately, because every varietal development goes through its own period of time, go through 5 to 10 years. So the every year we have a different set of environment, so that we should note down all the characters, all the uh, climatic parameters, how they are behaving, so that that is one kind of understanding, one kind of developing the existing cultivar.
Okay. And what are the potential economic benefits of farmers and agri entrepreneurs in adapting pre-breeding technologies?
That's what, ma'am. In general, without putting much efforts, let's say breeding means commercial breeding means they must go through all the steps, man. Pre-breeding means correcting the minor uh, defects with available materials existing in that particular area, and so that you can improve the yield as well as quality also with less inputs, so that finally you can reap the good benefits, man. Definitely, because commercial breeding is absolutely depend on pre-breeding. Any crop. Without pre-breeding, there's no commercial breeding, man. So that if it is there means there is a narrow genetic base. If it is there means broad genetic base.
Okay. And how do pre-breeding approaches contribute to environmental sustainability in agriculture?
Yeah, ma'am. Actually, in this case, because we are using wild kind of things and a wild array of germplasm, so that farmer, uh, the one variety which is adapted in one state or one environment, it may not adapt in different another environment or environment A, B, C like this. So that we can identify the materials, we can identify the same materials, or we can test the same entries in environment A, B, and C. So that depends on the environment we can identify. So that what happens, region specific or particular location specific entries we can develop, so that more genetic diversity is there, so that we can exploit the heterosis in broader genetic basis. That is, farmers may not know in our terminology, but we are in one of my slide. If you see, there are few major crops. They're confined to with minimum this one, ma'am. What you call minimum this one, minimum in varieties only. That leads to narrow genetic base. So that by using this pre-breeding approaches, we can narrow genetic base too, we can broaden the genetic base, and so that it can be sustainable for forever.
Okay. And what role can genetic resources play in pre-breeding efforts, and how can farmers and agri entrepreneurs access these resources?
Yes, ma'am. Any pre-breeding program start with plant genetic resources. Fortunately, we have a institutes like NBPG, National Bureau for Plant Genetic Resources. They maintain a good amount of germplasm for all the crops, so that we can bring it from them, and as per our requirements, as per our traits of interest, and that we can cultivate with the different blocks of the different locations or different regions, and we'll make it steady, and we'll show it to our farmers, so that each region they will have their own requirements, so that they need not depend on others. They can have their own program. Let's say if it is good at fertile environment, yeah, we can rise there itself, and we can produce the mass seed production, bulk seed production. We can distribute there itself, so that what happens? They should not. The most critical input is seed, ma'am, when it comes to different inputs. Not only chief input, but critical input, so that as far as plant genetic resources provide the good source of materials, that leads to the the basic step for the pre-breeding and as well as the commercial breeding program.
Okay. And are there any specific pre-breeding approaches that are particularly relevant for small-scale farmers or those in developing countries?
Yes, ma'am. In case of pulses, ma'am, actually, if you see PGNP, okay, actually, we have a very demonstrated capability to develop the varieties with existing germplasm. If we see in states like Andhra, Karnataka, Telangana, most of the entries developed from like registration like Long Farm Guntur, which is come under Acharya Engineering University, so that they are ruling forever. So that I can say that pulses are the good example for supporting in your question.
Okay. So now we have the last question. What are the current trends and future prospects for pre-breeding in sustainable crop improvement?
Yes, ma'am. Actually, as you know, in most of the crops, that genetic base was very narrow, man, because we are using the same entries. There are parent is common, ma'am. If you see the different cross combinations or different hybridization programs, we are using limited entries. That's what I'm keep on saying in my presentation or in so many questions what I have answered to so far. We should utilize as many as possible, more number of germplasm or whatever available for the particular crop, so that we can improve the diversity. If there is good diversity, we have a n number of variety, so that if one goes wrong, we should not wait for until next variety should come. As you have more number of diversity, so that if one fails, other is there. So that more number of diversity means we can develop trait specific variety for each and every entry.
Okay. So yeah, now we have the final question. Like, can you let us know a little about your career background and what made you to take an interest in agriculture field?
Yeah, ma'am. Actually, myself, Satish, basically I'm plant breeder, ma'am. I'm working in ITC. It's almost 15 years. And actually, any agriculture graduate, before I joining this one, of course, ma'am, my father is also farmer. But I thought that at that time while I'm joining, just like that, so that it's for sake of degree, I thought my father was joined. But once I joined, I realized how much important it is. Why 80%, 70 to 80% of the population of the country are depend on this one. So what are the infrastructure we have, and what are the different institutes were there? So that I thought that we have a nice platform so that I can explore. Then that leads to me to continue my agriculture science, agriculture graduation. And during my agriculture graduation, I thought that this genetics and plant breeding is the very interesting domain, so that we can deliver whatever required. that depends on trait of interest, so that we can use different biotechnology tools, statistical tools, so that it should be proven. Like I'm not criticizing other domains, ma'am. I'm not undermining the other domains, but they are entirely dependent more of operations and some observations. But it is not like that, ma'am. We should keep on observe. We should keep on raising the generations. It's like one should have a patience, ma'am. Plant breeding is not only simple science; it is art also, and one must have a patience, ma'am. That's what in one of my slide we mentioned that genotypic into environmental managemental activities interactions are highly required. So that leads me uh, to interest in this domain, ma'am. And I was delivered good number of varieties and hybrids in crops like tobacco and chilies, as well as my contribution in oil palm is there. So because releasing a variety in oil palm is a highly excellent task because it's a perennial crop. That pre-breeding work what I have done 10 years back, that work was fulfilled. And then now they are released as a different varieties in case of oil palm also. And we are in the first in India too, in case of tobacco, released the first CMS based hybrids, and that can rule the entire Andhra and Telangana, sorry, Andhra and Karnataka crop regions. It almost shares 92 95% of the entire plantation area of yeah, Andhra and Karnataka. Those
Okay.
are good breakthrough in case of my career.
Okay. Yeah. Okay. Okay, Dr. Satish, I don't see any more questions from our participants. So now we have come to end of question round. On behalf of agricultureinformation.com, we like to thank you for a detailed presentation and answering all the questions. And we also like to thank all the participants for joining this meeting. The meeting will now be closed.
Right. Thank you. Thanks a lot. And once again, I thank you to agricultureinformation.com. And because of some technical glitches, if there is any disturbance there, kindly apologize to me. And definitely, I will looking forward to give more number of presentations on different topics, with based on your prudent
Sure.
interest.
Yeah. Yeah.
Thank you.
Thank you so much. Sure. Yeah.